Let's have a look at this process called protein synthesis. So protein synthesis is simply the process where we make proteins. So you can say the manufacturing of proteins, etc., but it all means the same thing. So it's not a very complex process. It is one of those ones that you want to learn and just learn it the way it is and essentially regurgitate it onto a page. Towards the end, when we're looking at the last step over here, there is a little bit more understanding that needs to come in. But in terms of stating the two processes, they are actually quite simple. So protein synthesis consists of two main steps. The first one is called transcription and the second one is called translation. Now, the way I remember which one comes first, normally I try to do it alphabetically, but since they both start with a T, they both have an R and then an A, etc. So they both have trans as the beginning of their word, but transcription has a C and translation has an L, and C comes before L. So that's how I remember transcription comes first. This diagram here shows you the process of transcription, which in shorthand is just making a copy of the DNA code in the form of mRNA, and then translation is over here outside of the nucleus where you are translating the code on mRNA into an actual protein with amino acids joining. For the sake of this video, we focus on transcription first, but it's always good to come back at the end and look at this diagram to see how the process of transcription can differ in different diagrams. So when studying the process of transcription, it's the first step of protein synthesis, it's important that you note the similarities between this process that takes place in the nucleus versus the process of DNA replication. So I will try and emphasize where there are similarities and where there are differences, because those are the parts that are going to trip you up when answering a question like this in exams, if you're not aware of how similar these processes are. But at the same time, you can use this to help you and say, well, they are quite similar. I can study the process really well and then just learn what are the differences and where must I change the wording. So transcription, just like DNA replication, takes place in the nucleus. You can see this here. This is the nuclear membrane that is wrapped around the nucleus with pores in various places. And outside the nucleus is the cytoplasm of the cell. Transcription starts the exact same way that DNA replication does, with the DNA double helix unwinding. So when the DNA double helix unwinds, sometimes we'll show it fully unwinding like I'm showing you here because it's quite a short portion of the DNA molecule, or if we refer back to the original picture, it only partially unwinds. Then the next step is that these weak hydrogen bonds between complementary nitrogenous bases will break. And again, that is identical to DNA replication, and this will cause the DNA molecule to unzip, which forms two strands. Now, so far, this is identical to DNA replication. The DNA double helix is unwound, it is unzipped to form two strands. Here, I'm not showing you that it fully unzips because let's just say that this is the gene that codes for a particular protein or hormone to be made, and you only want to expose that gene. So most of the times, photos showing transcription will only show you part of the DNA double helix being separated from the other complementary strand. But the very key thing here that makes transcription different to DNA replication is the fact that only one strand acts as a template. That is huge. So the code for the protein is only found on this strand. Whereas if you go back to the video on DNA replication, you will see that both strands act as a template. Here, only one strand acts as a template. Now, the other thing that is different is the molecule that is being formed. We spoke about complementary base pairing when it came to DNA replication to form two identical DNA molecules. But here we're forming an mRNA molecule. In DNA replication, we used free DNA nucleotides from the nucleoplasm. In transcription, we use free mRNA nucleotides from the nucleoplasm to make a complementary mRNA molecule. So how this happens is that the free mRNA nucleotides that are floating around in the nucleoplasm looking for some way to go, they will now find their complementary base pair. Adenine will bond with thymine. Where there is adenine on DNA, however, like here, there's an adenine on DNA, RNA does not have thymine. So instead of thymine joining in here like it would with DNA replication, you get a new nitrogenous base called uracil. So that uracil will now come and bond on with the adenine on DNA. So it's the same type of shape and it matches complementary, but you only ever have thymine in DNA. You do not have thymine in RNA, instead you have uracil. That is the only difference between the nitrogenous bases. Then guanine will bond on with cytosine, guanine will bond on with cytosine, adenine with thymine, and cytosine with guanine. Remember that with these RNA nucleotides, you have a nitrogenous base, a sugar, and a phosphate. But the sugar in this case is going to be ribose sugar. Then what happens is that the nucleotides will all bond together to form a mRNA molecule. Now this mRNA molecule carries the code for the protein that needs to be made. So once this mRNA molecule has formed, it simply will now take the code to make the required protein. So we say it contains the coded message to make the required protein, and it will travel out of the nucleus and go to the cytoplasm on to the next step, which is translation.